In experiment 16, we learned about a technique that scientists have developed for separating a collection of different molecules. Let's look at another technique that scientists have developed for separating a collection of different molecules, and that technique is called crystallization. Here's the question that we want to answer in this experiment, and that is, can a mixture of water molecules and food color molecules be separated by freezing? Here's the experiment. Let's take a third of a cup of water, and let's add it to a paper cup. And now I'm going to add a single drop of blue food coloring to my collection of water molecules. And I'm going to take a spoon now and stir my collection of water molecules and blue food coloring molecules. And I now have a mixture of different molecules. Let's take our paper cup containing our mixture of different molecules, and let's place it in the freezer, and let's freeze the mixture. Now this will take several hours. Now I've already frozen a mixture of water and blue food coloring, and let me show you the results. If you look very carefully here, you can see some regions in the ice cube that's formed, some regions that are clear. These are regions where there are very few food coloring molecules. Now, if you compare this with our mixture that we put in, you can see that the water that went into the freezer is evenly colored. But the ice cube that forms upon freezing is not evenly colored. There are regions that are clear. Now, I believe this will show up more clearly when I tear the paper cup away, when I tear the paper cup away from our ice cube. Now let's look very carefully. Notice on the top of our large ice cube, you can see right through it. There are very few blue food coloring molecules on the top. Look down around the sides of our large ice cube as well. Again, we see regions that are very clear. Very few food coloring molecules are in these, are on the sides. Notice that most of the blue food coloring is concentrated in the center of our large cube, and also on the bottom of our large cube. Let's see if we can explain how the separation of blue food coloring molecules and water molecules occurred in this process. And let me use this setup here to demonstrate the formation of an ice crystal. I just have some wooden blocks, and we're going to represent a water molecule with one of our blocks. And we're going to represent a blue food coloring molecule with a lemon. Now, when an ice crystal starts to form, individual water molecules start to pack very closely together. Now, notice that all of our water molecules have the same shape, and so they're going to pack very nicely together and very close together. Now, let's take our lemon, which represents a blue food coloring molecule. It has a shape that is different from our water molecule. When it comes in and approaches our crystal that is growing, notice that it cannot pack very closely or very well in our crystal. So the blue food coloring molecule moves in, but it does not pack. Instead, another water molecule moves in, and takes its place. And our crystal continues to grow with more water molecules moving in. But eventually, the blue food coloring molecules have to become part of the ice crystal. And that is why we see most of the blue food coloring in the center of our large ice cube, because that's the part of the ice cube that freezes last. The ice crystal starts to grow first on the top of our water, and that's where we have our clear regions, and then along the sides. Can you understand now why this technique is called crystallization? It's because we are building crystals in the process.